Better? That's much better. Sorry, everyone on Zoom. Well, you could just restart it here anyways. So really this comes from like a really old interest of mine to produce a cognition producing system. The idea there is I create some universe in a computer and I press play. And after a long time, I look at some corner of it and there's something in there that's thinking. It's just something that interests me as a kid, and I've been wanting to do this for a long time. But I don't want to wait for like a whole brain or whole civilization to grow. So what's a simple organism that displays cognition? Well, slime molds. They solve foraging problems. They're the single-celled thing, which is really, really simple, really well studied, and have really cool behavior. So can I produce a system that creates a slime mold from scratch? Now, I was talking about this last year as well. This is kind of an extension of that work. But I have some rules on this. I don't want to just copy a slime mold because then I'm not really producing something from scratch. I don't want to train it because that's also just kind of copying something. And I don't want to wait forever. So I don't want to like define a Ruliad or like a primordial soup, a Big Bang, and wait for a billion years and in some strange corner it's tiny. I want to be able to like constrain the space and say, in this specific environment, I want this kind of behavior. And I want to be somewhat assured that I'm going to get something interesting and something that I can play with, something that runs fast, that I can move around quickly. So as a result of all that, I created the system. And just to give a brief overview of how it works, the main thing is that there's a substrate. So there's like, imagine like the Petri dish. And on that, there's each cell has values to it. So it's like how much nutrients is on that cell, is there energy on that cell? And this is represented as a big tensor on the GPU. And then that substrate also has a population that's beside it. And that population is a set of programs that run on that substrate. So each cell has a program assigned to it, you can imagine. Those programs sense and then act on their system, which is then passed through physics to say that's not possible or that has this effect. You visualize it, interact with it, and then you repeat this whole process. And so these organisms, these little programs, what are they? Well, they're hyper neat organisms, you could say. So that's like a compositional pattern producing network, this thing developed by Ken Stanley for neat, which is a bunch of little functions that work together to produce some value. And you use those to produce the values of a larger network, so a dense network. And there's a couple things about these. You can really quickly mutate and merge them. So you can just like change the structure of this CPPN, the small one, and that changes the larger structure through indirect encoding. And also, these are good at solving structured problems because they produced a structured feature of weights in the larger network. So like if you're solving chess, there's like a structure to the domain. So that's good. And for this problem, you can think of a cell, there's a left and there's a right as kind of a structured space. So that's the intuition behind using this. So for each little patch of cells in this grid, you take all of the sensors, pass it to this large network, and get some action space. You then take the action space, which you can define however you like, and apply it to the entire substrate for all live cells, which then passes through physics to produce the next step of the entire substrate. And the physics, this is kind of a cool point here, is the physics can use the mutate and merge operators of those CPPNs. So what that means is the network itself can mutate itself. It can say, I have one actuator, like my left arm mutates my neighbor. So that's something that you can program in Coralai pretty easily if you like. And then finally, you visualize that whole system, change the genomes if they're changed, and then repeat the whole process. So that's like the fundamental system that I built. Is kind of like something that you can install in pip eventually, not yet. And on top of that, I wanted to run an experiment to demonstrate it and explore it. And this experiment was just, I kind of built the system and I had to end my degree. So I put this together and started exploring it. So unfortunately, I don't have a rigorous analysis, but I'll tell a story about it at least. And this experiment is designed after slime molds. And so the channels, that substrate, is made out of three main components. One is energy, which you can imagine as like fluid, something that flows more. The other is infrastructure, so it's like walls, something that's a little bit more solid. And the third is communication, which is just whatever the network wants to use it for, like the hidden channels of a traditional NCA. The organism can sense all of that. There's nothing hidden there. And the actuators are invest and liquidate, which exchange energy for infrastructure, and explore, which is going to a neighboring cell, like which cell do I want to send some infrastructure to, and then updating the communication channels however it wishes to do that. The physics, the first little bit of physics is making sure that you don't ever lose mass or energy, just making sure that it sticks around in the right places. And then if two things try and go into the same cell, or four things go into the same cell, you spread the genome of the one that sent the most into that cell. So if you're all competing for space, if something's really high priority, then the biggest one wins. And then you also have some basic kind of like forest floor physics that I added in just for the fake ecology here, which is energy kind of spreads out. You have a day-night cycle, so you just add energy to the environment and remove it from the environment in like a sinusoidal manner. And you have like this falling debris, which will make sense in the next slide, and radiation. So radiation is just my poor man's way of adding mutation in there to get some evolution. And it actually worked kind of well. So you can see this is the code for setting up an experiment, not the physics. That's just kind of an example of Tai Chi. But on the left there, that's defining pretty much the entire structure of this experiment. So it's really easy to set up different channels and set up the kernel and so on. So if you just want to use like a Moore's neighborhood, you can do that. If you want to use multiple neighborhoods, you can do that. It's a very flexible system. This is really built for me to explore problems that I wanted to explore. And then if you want to write the physics, it's all in Tai Chi, which is this great language for Python. It can help you write really very efficient programs for the GPU and really easy to read code. And the result of this was I could run a 1024 by 1024 environment with 512 organisms at real time 20 FPS on my MacBook, which was a lot better than last year, which was on the CPU and very slow. So that was nice. And so now I'm just going to kind of tell a story of what I observed when I played with this system, like what came out of it. And so this will be kind of me enforcing what I see in it. And I found a pretty wide diversity of results here, which was exciting for me. And some of them kind of displayed what seemed like ecosystem dynamics and some very slime moldy dynamics too. And I really apologize for the squares. Those are supposed to be the ablations, like imagine a rock falling a slime mold and erasing it, or a tree falling with a bunch of energy. It's a square, it's not, it's really bad. And so this first little experiment here, you can kind of see, so green is infrastructure, red is energy, so that like the orange is like a piece of wood, it's energy. And the blue represents what genome, what program is running on that state. And sometimes the blue doesn't show up, also just my bad. But you can see there's like a little tumor there. That tumor is a mutation that happened. You can see it's kind of sticking around and it spreads around. And that really big patch of blue is a tumor or like a mutation that spread downstream from this organism. So you can kind of see that's the ablation there. That's the energy added to the falling trees. You can see that it really does look kind of like an ecosystem, kind of like slime molds. So on the first try, it kind of worked pretty well, which was cool. You can see competing organisms here. We also get two behaviors here, which you've seen slime molds, which I really didn't expect to emerge that easily. And they're not really doing the same thing as biology, but it's just interesting that came up. One is sporulation. So at some point in a slime mold's life cycle, it consolidates itself into all these little balls, and those balls turn into spores. And you see this happen here in this slime mold, where it's this network. It's kind of not a great network, but it's a network, and it turns into these balls. And it seems like kind of a natural behavior for this kind of physics here. You also get these little branching organisms that explore the space pretty well. They don't form a great network, like a really high efficient network. So it's not a great brain. So I'm not sure I really succeeded in my goal, but it's not bad either. So I played around with this a bit more. I removed the center of the environment and removed one half of it and saw what happened. And there's a little story I'd like to tell about the left one there, which is some organisms start to explore that space, but they use a lot of energy to do it. So that red is energy that's kind of being sapped out of the edges, or it seems like it's being sapped out of the edges. And during the night cycle, because they're not very efficient, they go extinct, all those ones that explored that area. And when they go extinct, all the ones from the edges can thrive, and you get a little bit more diversity in that center space. So I think that these are interesting dynamics that should be explored a little bit more. Seems like a promising system to me. It's another experiment I ran, and I'm just kind of modifying the parameters a little bit or just rerunning the same system, different initial conditions, and getting completely different results. So I thought that was interesting. And here you have a system with all these little branching organisms kind of co-evolving with these congealed organisms in here. So you can see there's one's looping, one's branching, these congealed ones. And it's just kind of, it stays like this forever, as long as I ran it. But it doesn't really do anything interesting. So I kind of made the conditions a little bit more extreme. So this one, I just added a bunch of energy during the day. And what you got as a result is this sort of fire-like organism, which spreads through the entire environment really quickly, but outcompetes everything else. So you can see like something tries to survive there, and then it dies. And in the top left corner, I'm not sure you ever see it in here, you get something that looks like it's going towards dense nutrients. I'm not sure if it's a selective search or not, but it certainly seems like something's happening. It's trying to survive. And so I thought of criticality at this point and said, okay, if there's a lot of energy and you get this dominant behavior, what happens when I remove all the energy? Well, nothing survives. What if I kind of try and play around with the energy level and try and find like a critical point? And at some point you get organisms which are behaving in a very similar manner, but you end up getting multiple types interacting in the same world. And you can see here that in that top left corner, you'll see an organism that's all branching right there with a hole in it. And right now it gets infected by another organism. It infects it, it eats it up, and then it spreads and it starts eating other organisms. And you can see this kind of competitive dynamic emerging from it, which I thought, again, was really promising for the system. So this was all just exploratory, right? I built the system and I played around with it. And what do I do next? What was happening next here? Well, firstly, more exploration, because it was a lot of fun. This all came from 3 days after I built the system of just playing around with it before I had to write my giant thesis. One really important thing that's lacking in all of these is obstacles. So anything can grow across the entire environment, which means you don't get niches, which means that you only have like one kind of ecosystem and one, that's a big limitation. Another is, I'm not really sure about Hyperneat. Honestly, I'm not sure it's the best. It's kind of old. I don't know. I'd like to play around with JAX a little bit more in developmental networks to have the organisms developing as they're alive in the ecosystem. I also think that it's going to be limited because you're kind of stuck at one level. Everything's local right now, and you can't really build, you can't take an entire area and have it act as an organism. It's all kind of distributed. So I think that hierarchical NCA have a lot of promise for this. So if you could have an organism kind of collect itself and then start controlling a larger environment, I think that could produce systems of systems, which would be promising. And a couple of things that I implemented, but I didn't have the chance to really analyze, was looking at the system with multi-scale complexity, which is like kind of going through the different renormalization groups of the system and saying, is there complexity at the small scale? And does that end up, is there still complexity if you kind of blur it at a larger scale? And if you keep on doing that, is there complexity at all these different scales? And for me, why I'm interested in that is it might signify systems of systems, or that you've reached criticality. It's not that it's not homogeneous and it's not chaotic or crystallized. And then phylogenetic diversity, just basic stuff you do with an ecosystem. And then eventually putting this on pip so everyone else can explore it. And speaking of clean code, this is very similar to a project that's being presented at 1500 in the same session, Biomaker CA from Google, with JAX and everything. So I think we kind of did this in parallel. I didn't know this was going on until I already started building the system. But this is a little bit different, uses kind of different physics. And there's also tool Vera, which is going on in like right now. But I'd love to try and listen to the system, just something that Jack Armitage talks about, which I think is really cool. And then finally, in the next session, I'm giving a talk on kind of using similar ideas to create interfaces to help people self-organize. It's kind of a long jump between the two, but in that talk, I describe how the two ideas kind of evolved in parallel and ended up co-evolving in an interesting way. So if you like those images and you think interfaces are cool, then come to the ERA session at 15:30. That's all the time I have, so thank you.
Okay, we have time for questions. So there is one here.
Thanks, really great talk. Thank you. I'm going to be horrible and ask a horrible question. So you mentioned that your aim for this was that you wanted to find cognition within your system. How are you going to tell when you see that?
Yeah, good question. So last year, when I presented Einchasm, which was a very similar system, I actually went a lot further in the exploration of it. And so I had weather going on, I had different physics and so on, obstacles, I was creating environments and so on. And what I proposed last year was when you create a system like this, and because it's trying to produce slime mold-like behavior, we have ways of testing or saying that a slime mold is thinking or is representing the fundamental mechanisms of cognition. So because it's interactive, you can design little experiments and say, if I put 2 pieces of food in front of it, does it go towards 1, and so on. If you have 2 things that are separated by obstacle and one learns something, can you remove that obstacle and then have them talk to each other and the other thing learns it? So you can kind of do like biological experiments in this. I didn't even start to touch this with this system, but I think that down the road in 3 years or something, you could do something like that. This is a lifelong project for me.
I have a question here.
I'm just wondering what kind of slime mold do you mean? Because you have shown some videos where we have seen slime mold dictyostelium discoideum, and in some parts we have seen Physarum polycephalum. So what is your objective? Yeah. Because you mixed both, so I was slightly lost.
Yeah, so I've mostly been focused on polycephalum, Physarum polycephalum, which, am I messing up? The dicta, right? Or yeah, well, since I'm kind of starting from scratch and I don't really define. Sure. Yeah, I'm coming from computer science. So I don't have the background in slime molds, unfortunately. I looked at them and I was inspired, and I thought there's some things that they're doing with physics, pumping liquid around and using that to communicate, using that to solve problems. And I thought, that's inspiring to me. I want to do something with that. Let me design a system that goes in that direction. So, sorry.
We have time for one last quick question.
Hi. So, I mean, first of all, I think Tai Chi